Oh, no.
I think we're live. Just check the stream. I'll wait for it to load. Yep, we're live. Kia ora tātou, ko Andrew Sargent tōku ingoa. I am a physics teacher at Te Aho Aotearoa Pounamu, and I'm here today to present our guest speaker, Amy Rowe. She will be going over the Life Processes Level 2 bio exam. Amy Rowe is a bio teacher at Wellington College, I believe that's correct.
Wellington Girls College.
Wellington Girls College, well, apologies. These webinars are brought in partnership with Study It. So at the end of this, if you have any questions, you can also always go down to the into the description and go to Study It from there. Amy's slides, I have edited the description in the video description, so you should be able to find the slides that she'll be going through. If there's any questions, just put them in the chat. I'll just interrupt Amy and then sort of at a time that's appropriate. But yeah, so over to you, Amy.
Thank you, Andrew. Kia ora, everyone. My name is Amy Rowe. I am a bio teacher, and I've been teaching life processes at the cellular level and the other level 2 standards for a number of years. So this is the second year in a row I've done a NCEA webinar for this standard. If you do have, or if you have seen my webinar from last year, I will be using some of the same slides, but I have updated it with some different exam examples, in particular ones from last year's cellular processes exam. So hopefully you'll get a lot out of it. All right, so this can be a really big standard, and a lot of students think that it is enormous, and there is so much to learn, and you are not wrong. Out of the 3 externals, it probably is the one that has the most content to learn. In terms of what you need to know for your exam, you'll need to know about cellular transport, proteins and enzymes, respiration and photosynthesis, and DNA and mitosis. However, a lot of the stuff that you will learn for this, there's a lot of crossover in the types of exam questions you're going to get. So it's unusual that you would get just one question on cellular transport. It would usually be a question that combines concepts and ideas from cell transport with proteins, or cell transport with photosynthesis. And similarly, it's unusual that you would get a question which is purely about proteins and enzymes. You would usually get something about proteins and enzymes crossed over with something from respiration. So the questions you get will combine different elements of the standard together. So it is a really good idea when you're studying for this exam that you do try and cover the whole thing, unfortunately. Try not to just silo yourself and focus on just one area. But because there is quite a lot to learn for the standard, I will be moving through it quite quickly. I'm going to be we'll just do a quick overview of an introduction to the cell, and then we'll be getting straight into cell transport, and then the other topics from there. I have tried to include some example past exam questions so that you get a little bit more familiar with the type of things you might be asked and how you might formulate a response to it. But if you do have any questions as we go, please just pop them in the chat. All right, so brief overview. We're going to be looking or talking about animal cells and plant cells. We don't usually get many questions on bacterial cells and things like that, so it'll be comparing animal cells with plant cells. And as you know, hopefully you know, our cells each have a nucleus and a number of other organelles. So for our animal cell, we have a plasma membrane all around the outside. We have ribosomes, which are our site of protein synthesis. We've got mitochondria, the powerhouse of the cell. I'm sure you know that one. And the cytoplasm, which everything else is floating in. And in contrast to our plant cell, this isn't the diagram of a plant cell. They usually have quite straight, rigid cell walls because they have a cell wall all the way around them. And then inside that is their plasma membrane. Similar to our plant cells, we've got our nucleus, we've got mitochondria, we've got ribosomes, but we also have chloroplasts as well. We will be going through all of those organelles and learning a bit about what they do. So just as a recap, if we're to compare our animal cells with our plant cells, we know that both have a plasma membrane, which is responsible for controlling the movement of anything into or out of the cell. So we're talking about water, we're talking about glucose, gases, nutrients, and other substances. And the plasma membrane is basically the barrier that controls what goes into or out of the cell. In our plant cell, we've also got a cell wall made of cellulose. And this is responsible for giving our plant cell rigidity and structure. So animals generally have some form, animals have some form of skeleton, either a bony skeleton, a cartilage skeleton, or a hydrostatic skeleton, which gives the animal a shape, whereas plants don't have any kind of skeleton, but instead they have a cell wall in each of their cells, which helps keep their cells rigid and gives them shape. Because they don't have a cell wall, animal cells can be a huge variety of shapes, and they're often quite rounded, whereas plant cells can often be quite rectangular in shape because of the cell wall that gives them that shape. In both the animal cell and the plant cell, we know that they've got cytoplasm within their plasma membrane. This is the jelly-like fluid in which everything else, all the other organelles, float about. You've got your nucleus, which again has a membrane around it, and it helps separate and protect the nuclear material, the DNA, from the rest of the cell's contents. We want to try and keep that DNA locked away, safe inside the nucleus. Ribosomes are the site of protein synthesis, found in both animal cells and plant cells. Mitochondria, found in both animal cells and plant cells. We'll get to that later. And they're really, the mitochondria are the site of where ATP energy is generated, which is why it gets the nickname the powerhouse of the cell. Chloroplasts are an organelle which are responsible for photosynthesis, and they are found only in plant cells, so you won't get an animal cell with any chloroplasts in it. However, what is important to remember, and a lot of students do get mixed up with this, is that animal cells have only mitochondria, plant cells have both mitochondria and chloroplasts. All right.
Just a question on the chloroplasts.
Yes.
Someone asked, how come some trees need more chloroplasts than others? And what's an example of something that needs a lot of chloroplasts?
Okay. Chloroplasts have the site of photosynthesis, and plants that will have a lot of chloroplasts will be perhaps, I would say, trees and plants that need to gather a lot of light in order to perform photosynthesis. So, for example, shade-loving plants tend to have very, very large leaves, and those leaves tend to be darker in colour because they're packed full of so many chloroplasts. And the reason for that is those shade-loving plants, or those plants that live in shaded areas, are not going to be exposed to such direct light intensity as other plants. So therefore, they compensate for that by having lots and lots of chloroplasts, so that for any light they do gather, they're able to use that light energy for photosynthesis.
And there's another question. Do animal cells burst during osmosis due to the fact that they don't have a cell wall, therefore they can't withstand the turgid pressure?
Yes, absolutely. And I do have a slide on that coming up.
Okay, cool. And then I assume you've probably got a slide how light wavelength affects photosynthesis.
I don't have a slide on that, but I can remember to talk about it when we talk about photosynthesis in greater detail.
Cool, sounds good.
Awesome, thank you. All right, so on to looking at the plasma membrane, which is important for cell transport. The plasma membrane surrounds our animal cells and our plant cells. It keeps the cytosol, or the cytoplasm of the cell, contained, and all of the organelles contained, and it controls the movement of anything that goes into or out of the cell. So water, glucose, ions, gases, and other substances, nutrients, those sorts of things. The plasma membrane is said to be a phospholipid bilayer, which basically means it is two layers of phospholipids. And on the left-hand side here, we have a single phospholipid. So it has the head of our molecule is the phosphate group. It is hydrophilic, so it is water-loving. And then we have the fatty acid tail down here, the lipid portion, which is said to be hydrophobic, which means it's water-fearing. When our phospholipids all join together, as we see here in our diagram on the right-hand side, all of the phosphate heads group together with the fatty acid tails dangling down. This top part would be a single layer, okay, but we have a phospholipid bilayer. Here's the second layer down here, okay. So this is our top layer, layer 1, and down here is layer 2. And you'll see that what happens is our phosphate heads group to the outside of our bilayer, and the fatty acid tails group to the inside of our bilayer. What this means is that water and water-soluble compounds on the outside and inside of our cell are quite comfortable being right next to those hydrophilic or water-loving phosphate heads, and the fatty acid lipid tails in the middle here control the passage of things that might be able to pass through our lipid bilayer. Another term you might hear in regards to the plasma membrane is that it is a fluid mosaic, which sounds like a really strange way to describe a membrane. What this means is that basically all of our phospholipids here and all of the proteins that are embedded in it are free to move. So the fluid part means that everything is able to sort of flow and move around easily. If you can imagine a whole lot of polystyrene balls floating on the surface of a swimming pool. They're all sort of packed together, but they're able and free to move. And that is a good representation of our phospholipid bilayer. The mosaic part, so if we say that it is a fluid mosaic, the mosaic part means that our membrane is made up of lots and lots of different parts, lots and lots of different proteins and other molecules that take part in our membrane. So the yellow things here, the tiny little yellow dots, are our phospholipids forming our bilayer, and spanning most of our phospholipid bilayer we have these protein channels and other accessory proteins that are able to control the movement of things in and out of the cell. Now, this membrane is semi-permeable and selectively permeable. So this means that some things are able to pass through it and some things are not. That's why we say it is semi-permeable. It's also selectively permeable, which means protein channels and other carrier proteins help to control specifically which molecules move into and out of the cell. So they are quite selective about what molecules are able to move in and out through those protein channels. So that's why we say that the plasma membrane is both semi-permeable and selectively permeable.
Just one question regarding the plasma membrane. Someone asked, do we need to study about the transmembrane carbohydrates, glycolipids, and glycoproteins? I noticed that these weren't on your slides about the plasma membrane.
Most of those you're unlikely to get any sort of question about. The reason that we look at the plasma membrane is because it is very important to being able to explain the cellular process of cell transport, so both active and passive transport. So it's unlikely you're going to get a question about glycolipids with regards to cell transport.
Cool. And I think there's one other question that I'm a bit confused about. What do we select to come into the membrane? Do we choose water to come in? I'm not sure if that makes sense. Does that make sense to you?
Not entirely. So water is able to move through the plasma membrane through pores, so protein channels that water is able to move through called aquaporins.
Cool. I hope that's answered that.
Okay.
So that's it for now.
Yeah, fantastic.
Cool.
So just having a look at this diagram, there are, as I said, lots of different parts to the plasma membrane, and all of those different parts, rather than being sort of stuck together like a brick wall, we say it is a fluid mosaic. There's lots of pieces that are free to move around on the phospholipid bilayer, kind of like icebergs floating on the sea. So some things that you may notice in this diagram You've got cholesterol, which helps to stabilize the plasma membrane, reduce its permeability to water-soluble molecules. So water-soluble molecules are ones that are dissolved in water, and the cholesterol helps to prevent those just passing through the membrane willy-nilly, and helps to control how and when they pass through. We've got protein channels and pumps, which allow the movement of specific molecules in and out of the cell, and those are probably the ones that are going to be of more interest to us. So you're likely to get a question in regards to protein channels or pumps. Glycoproteins, often these are receptors for specific molecules, and they will trigger cellular responses when these particular molecules are detected. Probably not likely to get a question about glycoproteins. It's much more advanced biochemistry than we look at level 2. And then we have oligosaccharides, which are in our diagram here, the carbohydrate chains, and they're involved in cells recognizing whether a particular cell is part of the same organism and signaling between cells. So in regards to an organism cells recognizing other self cells and being able to distinguish whether they are self cells or foreign cells that may need to be destroyed. So cellular transport, we look at 2 types.
One more question about the cell membrane. You said that someone asked, what do you mean you can choose what comes into the membrane? Because you said we can choose what comes in since it's semi-permeable.
Okay, so we can't choose. The cell can regulate what comes into and out of the cell based on what is needed by the cell for different cellular processes. And that's where we have active transport playing a role.
Cool. And then someone else asked a question. In past papers, I don't see many transport questions relative to respiration, photosynthesis, etc. How in depth should our knowledge be for transport?
That's interesting because when I was looking through past exam questions, I found 3 or 4 questions that combined cell transport with respiration and photosynthesis, including some of the examples that I'm going to be looking at shortly.
Okay, and the last one. So how can we tell what comes into the cell? Oh, so we can't tell them what comes into the cell. Not sure if that makes sense.
No, I'm not sure it does.
So I'm guessing the cell, if it needs something specific, like a specific salt, it can activate channels that let those salts in. Is that correct?
Yeah, that's right. Yeah.
Okay, I think that's what the student was asking. Okay, that's it for now.
Okay. So cell transport is involved with how cells move substances into or out of the cell and through the membrane. We look at 2 types. We have passive transport and active transport. So passive transport doesn't require any energy to happen. The stuff is moving along the concentration gradient from an area of high concentration to an area of lower concentration. And the terms you probably have already studied in regards to passive transport are things like diffusion, facilitated diffusion, and especially osmosis. And in contrast, we have active transport, which is the type of cell transport that does need ATP energy to drive it. The reason it needs energy is because it is going to be moving stuff, solutes, against the concentration gradient. So it's going to be moving it from an area of lower concentration to an area of higher concentration. So it's packing it where there's already plenty of that solute. So it requires energy for that to happen. The ones we look at are protein pumps and also the processes of endocytosis and exocytosis. We will go through endocytosis and exocytosis quickly. We don't often see questions about those processes. It's more about protein pumps and protein channels for active transport, but I will go over endocytosis and exocytosis as well. But let's start with diffusion. So diffusion is a passive process. This is where particles will randomly spread out from an area of higher concentration to an area of lower concentration. And this sets up what's called a concentration gradient. So I don't know if you can actually see my cursor. No, it doesn't look like that works. Down here in the lower right-hand corner, you can see there is a high concentration of these blue dots. So if you can imagine at the start, we had all of those blue dots packed down into this lower right-hand corner. And then through the process of diffusion, they have started to spread out from an area of high concentration to an area of lower concentration. And they will continue to move down the concentration gradient from an area of high concentration to an area of low concentration until the concentration is equal in both cases or in both areas. So the factors that affect how quickly that happens, the rate of diffusion, are how much difference there is between the high concentration and low concentration. If there is not too much difference between the high concentration area and the low concentration area, then diffusion is likely to happen slowly. Whereas if there is a very high difference between high concentration and low concentration, you're going to see diffusion happening quite quickly, where a lot of these blue particles quite quickly move into areas where there are none. The size of the particles diffusing is also important. So smaller particles are able to diffuse faster than larger particles. Temperature plays a huge role. So we know, or hopefully you remember from level 1 science, that particles at warmer temperature have more kinetic energy, so they move around more. If those particles have more energy, more kinetic energy, they're moving around more, so they're going to diffuse faster as well. If the temperature is cooler, those particles have less kinetic energy, which means they diffuse more slowly. And linked to both temperature and size of the particles is the state. So if you think of solids, liquids, and gases, gases diffuse much faster than liquids and much faster than solids. So small uncharged molecules like oxygen, carbon dioxide, glucose are able to freely pass through the cell membrane, moving down the concentration gradient. So from an area of high concentration of, let's say, carbon dioxide to an area of lower concentration carbon dioxide.
Just a question on that slide. Will it always be the case in which diffusion takes place until it reaches equilibrium?
Yes. Yep.
Cool.
So diffusion takes place until it reaches equilibrium, at which point it's important to remember that once you reach equilibrium, those particles don't just grind to a halt and stop moving. They will continue to move around, but they will be passing through the membrane in equal proportions, maintaining that equilibrium.
Cool. And there's one more question. Do we need to draw, or should you draw diagrams to support your answers?
Drawing diagrams is always absolutely fine in your exam. However, in my experience from marking, you do need to explain and refer to your diagram in your written answer. So if you want to draw diagrams to support your answer, absolutely, that is fantastic. A picture paints 1,000 words. However, you should still explain what is happening in your diagram in your written answer anyway.
Cool. That's it for this slide.
Cool. So facilitated diffusion. As I said last time, things like oxygen, carbon dioxide, and glucose can quite easily and freely pass through the plasma membrane. However, that's not the same for all particles or all molecules. So things like ions and other molecules can't diffuse directly through the plasma membrane. Okay, this might be because they're too big to squeeze in between the phospholipids, or it might be that they carry a charge or that they are not water soluble, so they're not able to pass through the lipid tails of our plasma membrane. So instead, to diffuse through the plasma membrane, they need a protein channel or a pore, which helps them pass through the membrane. So you can see in this diagram here, borrowed from the amoeba sisters, on the left-hand side, we see diffusion, where we have our happy red little particles moving straight through the plasma membrane, squeezing in between those phospholipids and moving from the outside of the cell to the inside of the cell. On the right-hand side of the diagram, we have our little purple guys there. They're not able to move directly through the plasma membrane. They're not able to squeeze through between the phospholipids. Instead, they're moving through a pore or a protein channel, which is kind of like a door, which is very selective and allows some molecules to pass through it. And you can see that in this case, we have our purple molecules passing through that protein channel from one side of the membrane to the other. So these transport proteins are specific. They will only allow certain molecules to pass through. So there might be certain certain ions, or in the case of aquaporin, it will only allow water molecules to pass through the pore. They are able to transport large molecules or ions and molecules with a charge. So, for example, sodium ions and potassium ions. The particles still diffuse at random, and again, it's still happening from an area of higher concentration to an area of lower concentration. But instead of passing straight through the plasma membrane, they need to go through a pore first, or a protein channel. Because it's still passive transport, moving from an area of high concentration to low concentration, no energy is required, so no ATP is needed. And you can see in the diagram on the top right-hand side, we have our little orange triangles moving from an area of high concentration outside of the cell through the protein channel to an area of lower concentration inside the cell.
Right, more questions. Transport of ions like sodium and potassium, is it like the electron transport chain? I'm not sure what that means, but maybe you do.
No, it's not really like the electron transport chain. It's considerably different. What we're looking at here is the plasma membrane around the outside of the cell. So these are molecules needed for cellular processes, usually being taken into the cell, or waste products leaving.
Okay, hopefully that answered that one. With these protons, so can the cell choose, or how does the cell, or do you need to know how the cell chooses what proteins go through? I think that summarizes someone's question.
Yeah, I don't believe there's any choosing involved with diffusion or facilitated diffusion, remembering that it is just the passive transport from an area of higher concentration to an area of lower concentration. And perhaps when we look at some of the examples given in some past exam questions, it might be a little clearer.
Cool. Hopefully that answers that one. Does all diffusion take place through a semi-permeable membrane?
The cell membrane is semi-permeable, so yes.
Cool.
Sorry, I understand the question. No, diffusion is a general term for a process of anything moving from an area of high concentration to an area of low concentration. So I think the SciPad gives an example of if you take a can of Lynx body spray and you spray it in the corner of your science lab, those particles of fragrance are going to move from an area of high concentration next to the can out into the rest of the room. So diffusion is the name given to that process of particles moving from an area of higher concentration to lower concentration and spreading out until they reach equilibrium. However, for the purposes of the standard, we are generally talking about things that are going through the plasma membrane.
Cool. Hopefully that answers that one. Someone asked, are questions on facilitated diffusion common, or are they usually on osmosis?
I have seen both. Usually osmosis, but I have seen questions on facilitated diffusion and comparing that with active transport as well.
Cool. And then someone asked, for example, in a woman's stomach where there's a fetus, does oxygen diffuse into the baby? I think they share a bloodstream.
Yeah, they share a bloodstream and the placenta. I think with the standard, we're looking at much more on the microscopic level rather than.
So you wouldn't get asked that question?
Yeah.
Okay, don't worry about it. If you're not going to get asked it, you can say that as an answer.
Yeah.
So
okay.
You're not likely to get asked that as a question. Cool. I think that's all for now. There's 61 kids on here, so there's lots of questions.
Okay, great. All right. So since somebody mentioned osmosis, we're going to move straight on to osmosis. So osmosis, again, is a passive transport. It is diffusion of water through a selectively permeable membrane. Again, because it's diffusion, it's from an area of high concentration to an area of low concentration. Now, what trips people up a little bit is we talk about an area of high water concentration to an area of low water concentration. And if you've done chemistry, you tend to think of water as being the solvent. So we don't tend to think about the concentration of water. However, hopefully, when I explain it, it'll make a little bit more sense. So in the diagram here, we can see this is an experimental system set up rather than a cell, but we have a differentially permeable membrane down the middle here, and we have a solution of sucrose, which is just sugar molecules, dissolved in water. Now, This semi-permeable membrane here will only allow water molecules to pass through. It won't allow sucrose molecules. So you can see that the smaller water molecules are able to pass backwards and forwards through this selectively permeable membrane, but the sucrose, the sugar molecules, are too large and they get compartmentalized or restricted to just one side of our membrane. And this is designed to model what we see in a cell. The water is able to move freely through the pores in a selectively permeable cell membrane. Just a quick reminder, anything or something that is dissolved in a liquid we refer to as the solute. So in this case, we have glucose molecules being dissolved in water. And you might also have examples where you have salts or ions dissolved in water. The glucose in this case, or the salts or the ions, are the solute. It's the stuff that's dissolved. Whatever the solute is dissolved in is called the solvent, and often that's water, as we can see in this diagram. And together, the solute and the solvent will make up a solution. So in this case here, we have a solution of water and glucose. We have our semi-permeable membrane here, and you can see, hopefully, based on the size of the molecules, that only water is able to pass through our semi-permeable membrane. Oh, there's our solute, there's our solvent, and the whole thing is our solution. Okay, so when the water has few solute particles dissolved in it, so not much stuff dissolved in it, we say that it has a high water concentration or a high water potential. It doesn't have much else in it, so as a whole, more of our solution is water. So we say that it has a high water concentration and a low solute concentration. So for example, a saline solution is just a salt solution. And if we have noticing that the YouTube video is a few seconds behind my explanation. If we have a 5% saline solution, okay, this means that 5% of our solution is salt and 95% of our solution is water. If we compare this to a 10% saline solution, that means that 10% of our solution is made up of salt and 90% of our solution is made up of water. So if we compare these two, we can see that the 5% saline solution has a higher water concentration, because 95% is higher than 90. So it has a higher water concentration than a 10% saline solution. So it can be tricky to think about what do we mean by water concentration or water potential. Just means relatively how much water have we got in our solution. All right, so when we talk about osmosis and osmoregulation, we want to be thinking about the concentration of water on either side of a plasma membrane or our cell membrane. Cells are surrounded by water in some form or other, so they need to be able to control the movement of water across the membrane. Okay, we don't want all of the water moving into the cell all at once, or we get cytolysis, which I'll talk about in a moment, and we don't want all of the water moving out of the cell all at once, or our cell will get dehydrated. And so we have what's called osmoregulation, which is basically regulating the amount of osmosis that's happening. So osmosis, as I said, a type of passive transport. No ATP is required. It is all diffusion. So the concentration of solutes inside the cell is high. If the concentration of solutes inside the cell, our green dots, is higher than outside the cell, we call this solution hypotonic. Okay, or hypo means low, and tonic means or refers to the salt concentration. So we put our cell here into a hypotonic solution. Okay, there is a lower concentration of solutes out here than in here. What that means is there is a much higher concentration of water outside the cell than there is inside the cell. What this means is that through osmosis, all of the water outside the cell wants to diffuse into the cell. Okay, so because we have much higher water potential or water concentration outside of our cell, the water molecules are going to want to equalize that by rushing into the cell to even out the water potential. So if we put our cell into a solution with a higher water potential than the inside of the cell, water will move into the cell from an area of high water concentration outside to an area of lower water concentration inside. If this isn't controlled with our animal cells up here, we get what's called cytolysis. So this is where a whole lot of water has moved through our animal cell membrane into the cytoplasm of our animal cell, which has caused the cell basically to swell up to the point that it bursts or lyses. In contrast to our animal cell, we have a plant cell. Now, plant cells, you'll remember, have a cell wall around them. So when the water moves from an area of high water potential outside the cell, rushes into the cell through the cell membrane, what that means is it causes everything in the cell to sort of swell up and press against the wall of the cell wall. Kind of, if you can imagine blowing up a balloon inside a cardboard box. That balloon is going to press against the walls of the box, causing pressure. Now, what we call that in plants is turgor pressure. So the plant cell becomes turgid because it has swollen with water, and the contents of the cell, the cytoplasm and the vacuole, are pressing against the cell wall, creating this pressure and making the cell rigid.
Just some questions, well, a few backed up. Had a bit of a debate going about aerobic versus anaerobic respiration. Aerobic respiration produces 36 ATP and anaerobic 2 ATP. I'm not quite sure what that means, but you might know.
Yep, I've got some slides on respiration later. 36 to 38 ATP for aerobic respiration and just 2 for anaerobic. Yep.
Cool, we'll go over it then. Do we have to know the difference between cofactors and coenzymes?
No, not really. No, I wouldn't say so.
Cool beans. Someone asks, what type of cells are flaccid? Is that animal cells? I put yes, because plant cells have cell walls.
Plant cells can absolutely be flaccid.
Okay.
Yep. So on my next slide, I'll be looking at if we have a cell inside a hypertonic solution, and what will happen is the water will leave the plant cell, leaving the cell flaccid. But both animal cells and plant cells can be flaccid.
Cool. And last one, if there's too much turgor pressure within the plant cell, is it able to burst eventually?
I don't think so. Never really considered it myself, but no, I don't think so. The cell wall does help to protect and maintain the integrity of the plant cell.
Cool. I think that's it for now.
Cool. All right. So on to our next slide on osmosis. What we're looking at here, I'll just wait for the YouTube to catch up. There we go. If we have a situation where the concentration of solutes or stuff dissolved in our water inside the cell, in this case our little green triangles, is lower inside the cell than it is outside the cell, what this means is there's more water concentration inside the cell than outside the cell. Okay, the solution outside is hypertonic. So hyper meaning high, tonic meaning salt, which is the stuff that's dissolved basically. So in our hypertonic solution, the inside of our cell has greater water potential than the outside of our cell. So if we put our cell in a hypertonic solution, the solution itself has a lower water potential. What this means is our water molecules inside of our cell are going to diffuse through the membrane, through those protein pores, from an area of high water potential inside the cell to an area of lower water potential outside the cell. So they're going to go and try and spread out until they equalize the water concentration. What this means for our cell is that water is going to move out of the cell from an area of high water concentration to an area of low water concentration. The cell loses water and becomes dehydrated. So what we see up here in the top right-hand side is our animal cell. They will shrivel up. Okay, so instead of being nice, round, plump animal cells, when water leaves, those cells become dehydrated and they start to shrivel. Again, because our plant cells have a cell wall, instead of just shriveling up, well, I mean, they still do. However, what happens is we see the plasma membrane and the cytoplasm pulling away from the cell wall to create these bizarre star shapes inside our plant cell wall. So this yellow structure here, okay, that's the cell membrane with its cytoplasm, and you can see because that cell has lost water, it has tried to shrivel up as much as it could, but of course the plasma membrane is still stuck and bound to the inside of the cell wall, so we get these strange star-shaped cells occurring, and we say that the plant cell has become plasmolysed. Now, another word for this is the plant cell has become flaccid. So if you think about my probably plants on a hot day, if they don't have sufficient water and they're left sitting in the sun and they haven't been watered, the soil is very, very dry, you will see that plants will start to wilt and to fold over, and that is a result of their plant cells becoming flaccid and wilting because they have become dehydrated and lost water.
Some more questions. How much do we need to know about mitosis?
Quite a bit.
Quite a bit?
Yep. We have got that coming up later.
Cool. I'll go over them. Someone asked if there was a cell, if there was 36 ATP in a flaccid animal cell, would this form a donut shape? I'm not really sure what that means.
I don't think that makes a lot of sense. We're sort of, yeah.
Fair enough. Do we need to know PMAT? I'm not sure what that means.
You don't need to know the specific names for the stages of mitosis, no, but they may be helpful for you to keep the different stages clear in your mind.
Cool. And I'm assuming this relates to a cell. If it shrinks, it becomes flaccid, correct?
Yes. Yep, that's right.
And then do we need to know the respiration stages in detail?
In some detail, yes. And I've got that coming up too.
Cool. That's all.
Okay, thank you. All right, so ideally, or what we want for our cell is we want our cell to be in an isotonic solution. So there we go. When the concentration of solutes inside the cell and the concentration of water inside the cell is matched by the concentration of solutes and water outside the cell, we say the solution is isotonic. Water moves into and out of the cell randomly. Okay, it's still diffusing across that plasma membrane, but it's maintaining that equilibrium, still maintaining the same water concentration. Therefore, the cell remains the same size, and the water and solute concentration stays the same, both inside the cell and outside the cell. And that's our ideal situation for our plant or animal cell. So it's well perfused with all of the water and solutes that it needs. So here's an example of an osmosis question. I apologise for the gross picture. I think this one dates from about 2016, from the 2016 exam from memory. So it is about a lugworm, which is a worm that lives on the sandy shores where saltwater concentration can fluctuate or change slightly. In order to survive in this habitat, our lugworm needs to passively adjust the saltwater concentration of its body to match the surrounding seawater. Oxygen consumption remains constant during this process, and the question has asked us to explain how saltwater moves across the cell membrane in a lugworm via osmosis and facilitated diffusion. So here is a question where we've got both osmosis and facilitated diffusion. Someone asked about it before. So before we talk about the answer, there's some things to notice about this question. And they're trying to point you in the right direction. So you can see that it says to survive in this habitat, the lugworm passively adjusts the saltwater concentration of its body. So that word passively isn't there for no reason. The exam writers are trying to direct you towards coming to the conclusion that you must be talking about passive transport here. So that is a bit of a clue. They also mention that oxygen consumption remains constant during this process. Now, we haven't talked about respiration yet, but we know that, or hopefully you remember that aerobic respiration requires oxygen. Aerobic respiration produces ATP energy. And so if, for our lugworm, oxygen consumption remains constant, not changing, that's another clue that we are talking about passive transport here. We're talking about a type of transport that doesn't require ATP. If it required ATP, then our lugworm would need to be doing lots of respiration to generate lots of ATP to move these substances around. But those are a few clues just in the question that direct you to the point that we want to be talking about osmosis and passive transport. So we know that water moves across the lugworm's membranes by osmosis, okay, and the salt in the seawater will move by facilitated diffusion. So our salt is moving from an area of high concentration in the seawater to an area of low concentration inside the worm. Okay, so it is moving down the concentration gradient, and it's going to move through protein channels in the cell membrane. This is because the salt is not able to pass directly through the cell membrane because of its solubility in water. So it needs to go through a protein channel. In contrast, water is moving from an area of higher water concentration inside the worm to an area of lower water concentration or lower water potential in the seawater by diffusing through the membrane. These processes are passive transport. They require no energy, and they work together to ensure that the lugworm always has the same saltwater concentration as the surrounding seawater. So our lugworm is able to passively move water and salt around using both osmosis and facilitated diffusion to try and maintain the same concentration of the seawater that it's living in. Are there any more questions on osmosis or passive transport before we move on to talk about active transport?
Yep. Are the channels the same for both active transport and facilitated diffusion?
No, they're quite different. So with facilitated diffusion, you might hear them being called protein channels or protein pores, whereas for active transport, they're usually referred to as a protein pump quite often because they are going to be moving substances against our concentration gradient. So like a pump moving from an area of low concentration to an area of higher concentration. We're pumping our substances against where they would naturally diffuse to.
And one more. I don't get how the worm gets salt out because the concentration of salt is higher outside.
Let me have another look at the answer. So salt is moving from an area of high concentration in the seawater to an area of low concentration inside the worm, and it's going through protein channels. So if the concentration of seawater or the concentration of salt in the seawater is higher, then you're going to have the salt moving through those protein channels into our worm. The question mentions that the saltwater concentration can fluctuate slightly. So if the next high tide, the salt concentration of the seawater drops slightly, then we'll see the salt concentration inside the worm will be slightly higher than the surrounding seawater. So the salt inside the worm will then move out through those protein channels into the seawater. Remember, our lugworm is trying to match the same salt and seawater concentration as the water it's living in.
So it doesn't use the salt at all, like it doesn't metabolize or anything. It just tries to keep it at a constant level. Okay, so that hopefully answers that question. Do they look different on diagrams for protein pump versus channel?
Yes, they will do. They should do. There will be enough information in the question that you will be able to tell, or you should be able to tell, whether you are dealing with a case of facilitated diffusion or active transport.
Hopefully that answers that. Must we always mention the diffusion of the solute? Is it always just salt in seawater, or is there osmosis with land animals?
Yes, you do see osmosis inside the tissues of land animals. Again, it depends on the question. As long as you understand the cellular process involved of passive transport, the question will provide you enough information and context that you should be able to apply what you know.
Cool.
And then how do we know the worm is salty inside?
Yeah, I've seen that question. It's very cute. The question again is telling you that our lugworm is trying to match the salt concentration of the surrounding seawater. So when I've written this answer, I've talked about salt moving from an area of high concentration in the seawater to lower concentration inside the worm. It would have been fine to say the opposite as well. So if, as I said, if the concentration of the seawater or the salt concentration of the seawater drops, then the inside of the worm is going to be saltier. So the salt will move from inside of the worm through those protein channels to towards the seawater. Remember, our worm is just trying to match the salt concentration of the seawater.
And like following up on that, why is the solute/salt moving? Or is it just the water? Is it the water moving or the salt moving?
They're both moving, and I would gather that probably the exam writer wanted students to be able to describe both osmosis and facilitated diffusion. That's probably the answer for that.
Cool. And then last one, with osmoregulation in plants, what molecules diffuse?
So osmoregulation or osmosis, we are talking about water.
Just water?
Yep. So with regards to our lugworm question, the part that is osmosis is the water moving from inside to outside the worm or outside to inside the worm. The salt is moving by facilitated diffusion. So when you get a question about osmosis, it's water. It's water that's moving.
Cool. I'm pretty sure that answered the next sort of couple of questions.
Cool.
Yeah, that's it for now.
Okay, thank you. All right, so active transport. As I've mentioned before, active transport requires ATP energy. So we're going to have chemical energy in the form of ATP driving this process. And the reason we need that energy is because active transport moves molecules against a concentration gradient. So with diffusion, we see molecules moving from an area of high concentration and spreading out to an area of low concentration, whereas active transport sort of pumps molecules in the opposite direction. So in our diagram up here on the top right-hand side, we can see that these little green squares are moving from an area of low concentration outside the cell and packing them inside the cell where there's already plenty of little green squares. So it is moving against the concentration gradient. Just a quick note on ATP. Hopefully you've been told that ATP stands for adenosine triphosphate. So this is a molecule, adenosine, that has 3 phosphate molecules attached to it, as we can see up here at the top of our cycle here. Now, ATP, or adenosine triphosphate, is able to release energy when one of our phosphate molecules is chopped off or cleaved off. So we have adenosine triphosphate. This is our chemical potential energy or chemical energy. And when one of those phosphates is removed, it becomes ADP, or adenosine diphosphate. Di because there are now only 2 phosphates left. Now, later on when we talk about respiration, we'll talk about the respiratory process adding phosphate onto ADP to turn it back into ATP. It's kind of like refueling the car, if you like. ATP, adenosine triphosphate with 3 phosphates, has lots and lots of energy or possible energy that can be used. When one of those phosphates is chopped off, it becomes ADP, which is low energy. What needs to happen in respiration is we need to add another phosphate onto our ADP molecule to turn it back into ATP. So when ATP is converted into ADP, the reaction releases energy, and this energy is used to power protein pumps and all kinds of other cell processes as well. It moves molecules across the membrane. Just as we saw with facilitated diffusion, active transport is selective. It will move specific molecules from an area of low concentration to an area of high concentration. It's not just going to move anything, it's going to move very specific molecules from an area of lower concentration to an area of higher concentration. So in this way, the cell is able to control what is entering or exiting the cell. So active transport.
Just a question about the ADP. Yep. Someone asked, we don't need to know about ADP, but from the previous slide, it sounds like you do. Is it?
I don't think I've ever seen a question that's referenced ADP. I've included it there as an explanation to sort of tie ATP in with the process of respiration and to sort of explain how ATP is energy inside the cell.
Cool. And last one, how does ATP lose a phosphate? Someone answered when ATP is broken down for energy. Is that correct?
Yes, that's right. Yeah.
Cool, that's us.
Cool. So active transport is performed by ion pumps and carrier proteins or protein pumps, which are found in the cell membrane. So in our diagram on the top right-hand side, we can see this protein pump here spans across the phospholipid bilayer from the inside of the cell to the outside of the cell. What may happen, and I've got a couple of diagrams later as well, is our solute here, our little purple circles, are able to move into our protein pump. ATP is then converted to ADP, releasing energy, and that energy causes the shape of our protein to change. So instead of being open at the bottom and closed at the top, what happens is that protein changes shape to be closed at the bottom and open at the top, which means those little purple circles are now able to move to the outside of the cell. And you can see on this side of the cell or on the inside of the cell, the concentration is very low, whereas the concentration of purple circles on the outside of the cell is very high. So our protein pump is moving our purple circles from an area of lower concentration to an area of high concentration, and it's using ATP to do it. So our specific substances, in this case our purple circles, binds to our carrier protein. ATP binds to the carrier protein, and the protein is able to turn the ATP into the ADP, releasing energy, which causes the carrier protein to change shape and ejecting the substance to the other side of the membrane. So this is an example of how glucose can move across a membrane and also how charged ions can move across a membrane. And the thing to remember about active transport is that it requires ATP energy and that it moves substances against the concentration gradient. So here is a specific example of a protein pump. This is the sodium-potassium ion pump. I've just seen Abby's question. Does it just flip to the other side? Yes, essentially. So it is open on one side, and then when ATP binds and is converted to ADP, the protein just changes shape and opens up at the other end, closes the bottom end, and the particles are able to move outside the cell. So we have our sodium-potassium ion pump. These are really important for the functioning of nerve cells and action potentials and for how nerve cells work. And in this case, as I said, they are very, very, these protein channels or these protein pumps are very, very specific. So in diagram number 1 over here, you can see that the sodium ions are able to bind. Now, for our sodium-potassium ion pump, it is able to bind 3 sodium atoms, sodium ions. And when those sodium ions bind, our protein is now full, and it is ATP is able to bind to our protein, and ATP will transfer one of its phosphate groups onto our protein pump. Okay, so in this way, ATP is turned into ADP by giving one of its phosphate groups to our protein pump. This releases energy and causes our protein pump to change shape. So you can see in diagram 2, our protein pump is open to the inside of our cell where our sodium ions are bound, and after the ATP has released its energy, it is now open at the other end to the outside of the cell. When this protein pump changes shape, our sodium ions are able to be released from the inside of that protein to the outside of the cell. Now, our protein pump is now open to the outside of the cell, and it's got a space where it can bind very specifically to potassium ions. So the potassium ions are able to bind to the inside of our protein pump. When this happens, again, these two bind, it causes a change in shape of our protein so that our phosphate group down here is released. Our pump changes shape and again becomes open to the inside of the cell. So by changing whether the protein is open to the inside or the outside of the cell, it's able to move particles our potassium ions or our sodium ions across our plasma membrane. So when our protein pump changes shape, we open to the inside of the cell, as we see in diagram number 6 here, our potassium ions are able to be released into the cell. Cool. I'm just checking the questions. All right. I won't spend too much time on cytosis because we don't often get too many questions about this. It's more about the active transport and protein pumps, and you wouldn't necessarily need as much specific detail as we went over talking about the sodium-potassium ion pumps. The things you need to remember are that these protein channels or protein pumps are very specific, so they're going to bind specific molecules. They require ATP, which causes a change in shape of the protein and is able to move molecules from an area of low concentration to an area of high concentration. Another form of active transport is cytosis. So this is this is where processes are able to move large amounts of material into or out of the cell by actually changing the shape of the membrane. So either folding the membrane or binding membrane-bound structures to the outside of the membrane and ejecting things. So the two that we look at is endocytosis. This is a process where processes are taken into the cell. So into. Hopefully that sounds the same to you. And there are two processes you might hear about, which is phagocytosis or pinocytosis. And I think the SciPad and possibly the ESA study guide refer to phagocytosis as being where the cell eating or it's absorbing solid particles, whereas pinocytosis is talking about, or they refer to it as the cell drinking. This is where the cell is able to take in fluids or liquids. So what happens for both phagocytosis or pinocytosis is kind of the same. So we have our plasma membrane here. You can see our lipid bilayer. And what happens is when a solid particle, as we see on the right-hand side here, our large particle, presses against the plasma membrane kind of creates this dip or pit, or we say that it invaginates and then forms kind of like a little pocket or loop that is able to reform at the top here, kind of like what we see in the diagram here on the lower left-hand side. So you can see that these particles on the outside of our cell are causing this dip or pit in the membrane. The membrane then loops back around and reforms, okay, and sort of pinches off a little loop or a little what's called a vesicle, which surrounds the solid material that the cell has taken in. This is where the plasma membrane being a fluid mosaic is really important. So don't think of our plasma membrane as being a brick wall. It is fluid, it's able to move, and that's what's happening here. So our plasma membrane creates this dip, it invaginates, and then the two pieces or the two sides of our plasma membrane then squeeze back together and they pinch off this little vesicle, okay, which has our solid substance in it, surrounded by a little loop of, or a little circle bubble of phospholipid bilayer.
Just some questions. Is phosphate ion a cofactor? Do they need to know it?
No, they don't need to know it.
Cool beans. What role does it play in protein pump? Do they need to know that?
So the phosphate has to do with when ATP binds to our protein pump, it transfers one of the phosphates from ATP onto the protein pump. So in effect, it's turning ATP into ADP. And when ATP turns into ATP turns into ADP, it releases energy. So that is the energy that's released in order to change the shape of our protein pump.
Cool. I'm not sure if this is related. What is the function of the matrix in the mitochondria other than the Krebs cycle? Do they have to know that for level 2?
Well, the Krebs cycle is pretty important. So
yeah.
Stick with that. Yep.
I assume that you'll be going over the Krebs cycle later on.
Yeah. Yeah.
Yeah, okay, cool. What's the difference between a vesicle and a vacuole?
Vesicles are teeny tiny and vacuoles are huge. So it's more to do with the size. Vesicles, very small, move around inside the cytoplasm, whereas vacuoles tend to be almost more permanent inside the cell.
And do they need to know that for the exam at all?
Unlikely.
Cool. And that's it for now.
Yep, cool. All right, so as well as the process of endocytosis, where things are coming into the cell, we also have exocytosis. This is where substances and waste products are removed by the cell. So endo, into, exo, like exit. So this is where stuff exits the cell. So it's for removing waste products, or it might be important for cells that are excreting hormones or neurotransmitters, or anything that is produced by the cell that needs to go somewhere else. So substances are stored or packaged into a vesicle within the cell, which is bound up by a membrane. What happens is that membrane squeezes up against the cell membrane, and they sort of merge together. So the vesicle membrane merges with the cell membrane, and when it merges, it basically opens up the vesicle to the extracellular environment, so outside the cell. And as it does that, as it merges with the plasma membrane and stretches out, it releases its substances to outside the cell. Again, this is an active transport process, so it requires ATP to drive it because you're dealing with changing the shape of the cell membrane. However, you don't need to know too much about the details of that. No questions? Great. All right. So this is a question from last year's exam paper around osmosis and active transport. And hopefully if we go over this question, you'll get a feel for how much detail you need to provide in your answers and also how much you don't. So rawaru, or blue cod, is a marine fish that lives in salt water. Rawaru cells maintain a water concentration that is different from our surrounding seawater. So this is different to our lugworm. Our lugworm trying to match the concentration of our seawater, whereas rawaru need to maintain a particular concentration of water in their cells that is different from the surrounding seawater. We know that if water is moving, it is through osmosis generally. So water is lost by osmosis through the gills and through the skin cells. Our marine fish drinks seawater in order to replace the water that is lost by osmosis. So because the fish is losing water through osmosis, through its gills and skin cells, it's still going to need lots of water for all of its cellular processes. So the only place that our blue cod is able to get that water is by drinking seawater. However, when our blue cod drinks seawater, it's not just taking in water, there's a whole lot of salt in there as well. So marine fish use active transport to excrete sodium and chloride ions through their gill cells. So this question's quite nice, I guess, because they're giving you and directing you towards what you need to be talking about. You need to be talking about osmosis, and you need to be contrasting it with active transport. The question also says that seawater is made up of about 96.5% water and about 3.5% solutes. So solutes is anything that's dissolved in it, including sodium ions and chloride ions. The question asks us to use the information in our diagram above to discuss how rawaru maintain a water concentration inside their cells that is different from the surrounding seawater. In your answer, you should explain what osmosis is. So we're just giving a definition, and explain why it occurs between the fish and seawater. You need to explain active transport and why it occurs between the gill cells and seawater, and discuss why the rawaru must drink seawater, but they must also actively remove the sodium ions and the chloride from its body. So you may have gone through this in your classes already, but hopefully I'll be able to add something on there for you. So the first part of that question asks us to explain osmosis and why it occurs between the fish and seawater. So you would start out by just giving a definition of what is osmosis. Osmosis is the diffusion of water across a semi-permeable membrane from an area of higher water concentration to an area of lower water concentration. So you want to link that to the context of the question. So our fish, the water is being lost from our fish. So the interior or the inside of our fish's cells have a higher water concentration than the surrounding seawater. So water is moving from inside the fish's cells to outside the fish's cells by osmosis. Osmosis is passive transport and requires no energy. Water is lost from the fish out to the seawater because the fish's body has a higher concentration of water than the surrounding salt water. This means the fish's body has less salt in it than the surrounding salt water as well. The fish needs to drink the salt water to replace the water that's lost by osmosis, because water is still needed for all of the fish's life processes. However, when it drinks the seawater, it's also taking in the sodium ions and the chloride ions because those are dissolved in the seawater. However, these ions are not wanted in large concentrations, so they must be removed, which takes us to the next part of the question, which is to explain active transport and why it occurs between the gill cells and the seawater. And you'll notice from our fish diagram up here, it says that marine fish use active transport to excrete the sodium ions and the chloride ions through the gill cells. So active transport is the movement of substances through proteins in the cell membrane or through protein pumps in the cell membrane from an area of low concentration to an area of higher concentration. So it's moving those substances against the concentration gradient, and it requires energy or ATP. Sodium ions and chloride ions are taken in by the fish when it drinks the seawater, but the fish doesn't want to maintain a high concentration of sodium and chloride ions in its body, so it needs to remove those ions from its body. Because there is a higher concentration of sodium ions and chloride ions in the seawater than inside the fish needs to use active transport to move the sodium ions and the chloride ions from an area of low concentration inside its cells to an area of high concentration out in the seawater. Because of the size or the polarity or the solubility of these ions, they can't move directly through the cell membrane, so they can't diffuse through the cell membrane, and they must be actively pumped out through protein channels, again from an area of low concentration inside the fish's body to an area of high concentration to the surrounding seawater, which requires energy or ATP. Lastly, you might want to wrap it all up. So discuss whether rawaru must drink seawater but also actively remove the sodium and chloride from its body. So rawaru must drink seawater to replace the water that's being lost because of osmosis, and it needs that water for life processes. If the fish loses too much water to osmosis, then it will become dehydrated, it will shrivel up, and it won't be able to perform its life processes, and it'll likely get very sick and die. However, sodium ions and chloride ions taken in when it drinks seawater, so they must be actively removed from its body. These molecules must be pumped out through protein channels. If the sodium and the chloride are not actively removed, they're going to build up in the fish's body because the fish is going to continue drinking seawater to replace that lost water. And as those build up, eventually you're going to get a higher concentration of sodium ions and chloride ions inside the fish than you have in the surrounding seawater. What this means is you'll get a reverse of the direction of osmosis. So instead of the water moving from inside the fish to the seawater, you're going to get water moving from the seawater into the fish. And if it all rushes in to try and equalize that concentration gradient, then you can cause the fish's cells to swell up, burst, and die. And if you recall, a fish requires its gill cells in order to be able to exchange oxygen. If its gill cells swell up, burst, and die, then so does the fish. Any questions on that question?
Will the cell, or maybe the fish, will it become flaccid if not enough water? Not sure what that means.
Well, the fish would probably die before it became flaccid. Yes.
Cool.
Yeah.
I assume flaccid means the water's going out of the cell?
Yes. Yeah.
Cool. No, it's all good. That's it.
Cool. All right. So proteins and enzymes. There are many different types of proteins and structural proteins used, antibodies and enzymes that are important for the cells processes. Proteins are long chains of amino acids that are then folded into very, very specific shapes, and they're held in that correct shape by chemical bonds that form between the amino acids. And the shape and structure of proteins is vital to their correct function. Enzymes are proteins that catalyze chemical reactions. We know that enzymes speed up reactions by lowering their activation energy. They are biological catalysts. The enzyme is not changed by the reaction that it catalyzes, so it can be used many times. And enzymes are very specific for the reaction that they catalyze. So generally speaking, we say that one enzyme catalyzes one specific reaction. And if you're not sure if you're dealing with an enzyme, you can usually tell by looking at the name of the enzyme. They often end in the letters ASE or A's. And often they're named for the type of reaction that they catalyze. So, for example, lipase catalyzes reactions with lipids, which are types of fats. Protease catalyzes reactions with proteins, and lactase catalyzes reactions with lactose. So the A's part is a bit of a hint or a tip that you're dealing with an enzyme. All enzymes have an active site. Okay, this is a groove or a nook in the protein structure that the enzyme's specific substrate can fit into. The substrate is the substance that the enzyme is going to act on to catalyze the reaction. So it's going to usually turn it from one thing into another. According to the lock and key model, the substrate must perfectly fit into the enzyme's active site for the reaction to be catalyzed. And certainly the shape of the enzyme's active site is vital to the functioning of the enzyme. The substrate binds and is held in place by hydrogen bonds. The reaction is catalyzed, and then the products are released by the enzyme, breaking those hydrogen bonds. Another model of the substrate and active site binding that you will have learned is the induced fit model. So this is very similar to, or it's quite similar to the lock and key model. However, according to the induced fit model, the enzyme's active site is a loose fit for the specific substrate. So the specific substrate can bind to the active site, and when it does, the enzyme's active site changes shape slightly to better fit the substrate and lock it in place, kind of like giving it a bit of a hug. This change in shape of the enzyme causes a change in the shape of the substrate too, possibly putting the molecule under tension and making a successful reaction more likely. So the induced fit and the lock and key model are quite similar. The difference is with the lock and key model, the enzyme's active site doesn't change shape at all, whereas with the induced fit, when the substrate binds, the active site of the enzyme kind of hugs or locks on to the substrate, changing the shape of the active site slightly, which can lead to a more successful reaction. Yes, you should know about the two models, the lock and key and the induced fit model. So denaturation is when a protein structure or shape has changed so that it no longer works. If you've ever cooked an egg and you've seen egg white go from sort of that slippery clear egg white to white and firm, you have seen protein denaturation. So it's where the structure or shape of the protein has changed and it no longer has the same shape and no longer has the same function. So proteins, if you remember, are held in their structures by hydrogen bonds and disulfide bonds, and factors such as high temperature and changes in pH, either becoming more acidic or more basic, can interfere and break those hydrogen and disulfide bonds. If those bonds are broken, it's going to change the shape of your protein, and a loss or change in the shape means a loss or change in the function of your protein. So we have our folded protein here on the left-hand side. It undergoes denaturation. All of the bonds holding the protein in that structure have been broken, and we have this unfolded protein, which is now just a long string of amino acids. Denaturation is irreversible. We can't then change our unfolded protein back into a functional protein. All right, so just the summary, factors that affect enzyme function. things like temperature. So too cold, our substrate particles and our enzyme are not going to be moving around with much kinetic energy. They're not going to be colliding with the enzyme very often. So therefore, there's going to be fewer successful reactions occurring. At optimum temperature, our substrate particles are colliding often with our enzyme. Therefore, they're binding to the active site and the reaction is able to be catalyzed efficiently. However, if the temperature gets too hot, the enzyme can denature because the hydrogen bonds and disulfide bonds holding our protein together can be broken. This means the shape of our enzyme will change and the enzyme cannot catalyze the reaction. pH, most enzymes work inside the cell where we have an optimum pH of around about 7. If we get a change in the pH, so more acidic or more basic, this can affect the way the substrate binds to the enzyme or can entirely cause the enzyme to denature. And again, a denatured enzyme is not going to be able to fulfill its function. Concentration of the substrates is really important. If we think back to level 1 acids and bases again, when the concentration of our substrates or reactants increases, the rate of reaction is going to increase because you're going to have more successful collisions with our enzyme. If the concentration of substrates is low, then you're going to have less successful collisions of the substrate with the active site. Therefore, it's not going to be able to catalyze the reaction as quickly. And also we have inhibitors. So inhibitors are substances that are able to bind to the enzyme and prevent it from catalyzing a reaction. Now, the reason that I wanted to go through the factors that affect enzyme function is it's rare that you would get a question that's solely about enzyme function. You'd often get this linked in with possibly respiration or another cellular process. And the reason why it's important is respiration and photosynthesis and mitosis are all enzyme-driven processes. So they all happen because of the action of enzymes. So when you're asked about the factors that affect photosynthesis, there are going to be some that are specific to photosynthesis, yes, but a lot of them are going to be around the factors that affect enzyme function because photosynthesis is carried out by enzymes. Similarly, if you're asked about the factors that affect respiration, there are some things specific to respiration, but mostly it's going to be things like temperature, pH, and the concentration of substrates and whether there's an inhibitor, because respiration is driven by enzymes. So in terms of learning about how important these factors are affecting enzyme function, it's really important because you're going to be able to talk about that in regards to these enzyme-driven processes like photosynthesis and respiration.
We have a lot of questions, if that's all right. Are enzyme inhibitors permanent?
Some are permanent, some are not. So some will bind to the active sites, so they may be competitive inhibitors, and again, they may bind permanently or they may detach. If they bind to the active site, they inhibit the enzyme by basically filling up the active site so the substrate can't bind. And in some cases, they can then detach and the true substrate can bind. We have what's called a non-competitive inhibitor as well. So this is where a substance binds at a site on the enzyme, which is not the active site, so it binds somewhere else. But when that inhibitor binds to the enzyme and that other site, it causes a change in shape of the whole protein. So that change in shape of the whole protein means that the substrate can't bind to the active site anymore. And those tends to be more permanent inhibition.
Cool. Do we need to mention the induced fit model is more accurate? Question mark.
Yes.
Cool. I think someone asked, is a protein made up of enzymes? I'm pretty sure it's the other way around. An enzyme is made up of proteins. Someone answered that. That's correct.
Yeah, an enzyme is a protein, and proteins are made up of amino acids, so strings of amino acids.
Cool. How do cofactors affect enzyme involved in DNA replication? I couldn't understand the substrate-enzyme relationship in that process.
I would need to look at the specific question. Cofactors are generally things that bind to an enzyme to help it carry out its function, and the enzyme is not able to function and carry out or catalyze its reaction without the cofactor having bound to it. But I'm not really sure how that relates to the substrate-enzyme relationship.
Cool. Hopefully that's answered that. Will cofactors and coenzymes be included as factors?
Not generally. So cofactors and it depends on the question. It's unlikely to come up in a level 2 question. Cofactors and coenzymes affect whether the enzyme is able to perform its function correctly. But yeah, from the level 2 questions that I've seen, it doesn't come up.
And finally, what's the difference between a cofactor and a coenzyme?
So a cofactor is usually something like a vitamin or a metal atom that needs to be bound by an enzyme to keep it in the correct shape, also give it the correct chemical properties to catalyze its reaction, whereas a coenzyme is where you might have... it's been a long time, where you might have 2 enzymes that work together. So they're both required to catalyze a specific reaction.
Cool. Do they need to know that for level 2 though?
No.
Cool. And then hopefully, if they don't need to know this for level 2, just say it said that magnesium 2+ has acted as a cofactor for many enzymes involved in DNA replication. But how does it help enzyme activity? How do enzymes work in DNA replication?
Okay, so yes, magnesium is one of the metals that I mentioned that can act as a cofactor. It needs to bind to the enzyme for the enzyme to function correctly. So if the magnesium is not bound to the enzyme can't perform its role effectively, can't catalyze its reaction. How do enzymes work in DNA replication? So if you know the names of the enzymes involved, DNA helicase, the word ase tells you it's an enzyme. So DNA helicase works to unwind the DNA molecule. You've got DNA polymerase, which is responsible for linking together all of the nucleotides that are bound to the template strand of your DNA. So every process in DNA replication is controlled by a different enzyme.
Cool. I think that's it for now.
Cool. All right. Inhibitors, I think I have already explained this. So certain molecules can inhibit an enzyme from carrying out its job, so prevent it from catalyzing its reactions. They're considered poisons, and they can include heavy metals such as lead and mercury, and I think cadmium came up in an NCEA question a couple of years ago as well. So there's 2 types of inhibitors that I mentioned. We have site-competitive inhibitors and non-site-competitive inhibitors. So site-competitive inhibitors will bind to the active site of the enzyme. They block the active site so that the substrate can't bind. If the substrate can't bind to it, then the reaction can't proceed. And as I mentioned, these may be reversible or irreversible. So if this is where our normal substrate would bind, over here, our competitive inhibitor binds to it instead and blocks the active site so that our normal substrate can't bind to it. In contrast, we have site competitive inhibitors. This is where they bind to a location away from the active site and causes a change in the shape of the enzymes, the whole enzyme, so that the active site can't bind to the substrate. If the substrate can't bind securely, then the reaction can't proceed. And often our non-competitive inhibitors tend to be more permanent inhibitors. So you can see here the inhibitor, the blue inhibitor, is binding to a site on our enzyme, which is not the active site, but in doing so, it causes a change to the shape of the active site. So now our normal substrate might be in the active site, but it doesn't fit anymore. Okay, so our reaction can't proceed. It cannot catalyze the reaction. So enzymes and enzyme function are vital for the cell to carry out its cellular processes. And as I mentioned earlier, almost everything the cell does is controlled by enzymes. So DNA replication, truckloads of enzymes, photosynthesis, enzymes, respiration, many, many enzymes involved. So if you're asked a question about the factors that affect DNA replication, there will be some specific to DNA replication, but you can also include all of the things that affect enzymes, so things like pH and temperature and the availability of substrates. And as I've mentioned a couple of times, exam questions about enzymes will often be combined with a life process, such as photosynthesis, respiration, or DNA replication. Here is an example. So the eastern oyster's cellular respiration and enzyme activity are affected by temperature, oxygen concentration, and cadmium, a heavy metal. Discuss how these factors can affect cellular respiration and enzyme activity in the eastern oyster. You should describe the purpose of an enzyme, explain how temperature and cadmium affect enzyme activity, and discuss how environmental temperature, oxygen concentration, and cadmium can affect the rate of cellular respiration in our oyster. So the first part, describe the purpose of an enzyme, is just your straight definition. An enzyme is a biological catalyst that speeds up a chemical reaction by lowering the activation energy of the reaction. So we need to move on to our next step, which is to explain how temperature and cadmium affect enzyme activity. So this is just as we've been describing, those factors that affect enzyme activity. So respiration is a cellular process that's controlled by enzymes. So enzymes need to function at an optimal temperature. If the environmental temperature is too low, then collisions between the substrate and the enzyme will be infrequent. Therefore, the rate of respiration will be low. If the environmental temperature increases, then our substrate and our enzyme have more kinetic energy, so collisions between our substrate and enzyme are going to be more frequent, so respiration will also increase. The optimum temperature for our oyster will be the temperature at which the rate of respiration is highest. Okay, so this is where our substrate and our enzyme are whizzing around, having lots of successful collisions, and our enzyme is able to catalyze the reaction to produce a product. However, at a temperature above the optimum temperature, the enzymes may become denatured, which makes them inactive, and denaturation is irreversible. Denaturation occurs because of a change in the shape or structure of the active site, which can no longer fit the substrates involved in the reaction. Once the active site begins to change shape, the rate of cell respiration will decrease or may stop altogether, since the substrates can no longer bind to the active site of the enzyme, so the enzyme can't catalyze the reaction. All right, so our question also asks us about how cadmium affects enzyme activity. The question kind of points you in the right direction by mentioning that cadmium is a heavy metal. So cadmium is one of those inhibitors or poisons that is going to inhibit the activity of our enzyme. So cadmium is a heavy metal that acts as an enzyme inhibitor. It can bind to the active site of our enzyme, blocking it or changing its shape. This prevents the substrate from binding to the active site, and if the substrate can't bind, the enzyme can't catalyze the reaction. So this is going to prevent our enzyme from catalyzing the reaction. Our enzyme's not going to function correctly, so no product is going to be formed. All right, any questions about enzyme activity before I move on to?
There's just one question. What is does it mean cofactors can be reused for every reaction? Yeah.
They are usually required to be bound to the enzyme for the enzyme to function correctly, but they don't take part in the reaction. So the cofactors are generally just it's kind of like your car keys. You need your car key to start the car. So if driving the car is the reaction, you need your car key, which would be your cofactor, inside the car to make the car work. It doesn't usually get used up in the reaction.
Cool. And then what is the enzyme's role in photosynthesis?
Everything in photosynthesis is controlled by enzymes, but you don't need enzymes involved.
Yeah, that answers that. So heavy metals and poisons, they must be inhibitors.
Yes. Yeah.
Cool. And how do you know cadmium is an inhibitor and not a cofactor?
It's in the context of the question.
Cool.
So it tells you, that cadmium is a heavy metal, and you would be expected to know that heavy metals are generally inhibitors of enzyme activity.
Cool. I think that's it. It's 8:30 as well, so the time.
Yeah. All right. So I'll go on to respiration. I'm not sure. Well, I won't be able to get much further than that, I don't think. But if there are specific questions or specific processes that people want to go over, if they want to pop it in the chat, and I can try and go over it quickly. All right. The mitochondria is the site of aerobic respiration. This is where glucose is broken down to water, carbon dioxide, and produces lots and lots of ATP. So we're talking about aerobic respiration here. You quite often will get a question asking you to identify and name the different parts of the mitochondria. So on the outside, we have our outer membrane. This is kind of like the big bag that contains our mitochondria, and it regulates the molecules entering and exiting the mitochondria. Inside, we have our inner membrane, which is folded up like this, kind of like the wall of the gut, if you like, into what are called cristae. So each of these folds is a cristae. And these cristae act to increase the surface area of the inside of our mitochondria. Now, the cristae is the site of a whole lot of respiratory reactions. So we want to have lots and lots of surface area there, so we've got lots of space for those reactions to occur. The cristae are the site of our electron transfer chain reactions. So our electrons are passed backwards and forwards across our cristae membrane. So that's again why we need lots of surface area for them. And inside our cristae, or inside our inner membrane, we have the jelly-like filling called the matrix. And this matrix is just packed full of many enzymes. So this is where the Krebs cycle occurs. And if you go on to study bio at uni, you will look at the Krebs cycle in excruciating detail in level 200 level. And I think there's about 40 or 50 enzymes that you need to learn for the Krebs cycle, and it's very, very complex. So level 2, you don't need to know all of that. You just need to know that the Krebs cycle occurs in the matrix. So the role of the mitochondria is to produce ATP, and as we've talked about, ATP is used by all cells for energy, for active transport, for protein synthesis, for movement. Heaps and heaps of life processes require ATP. Cells that have very high energy demand, such as muscle cells or liver cells because they're secreting things, sperm cells because they're moving, all of these types of cells that have a very high energy demand will have lots and lots of mitochondria in them. And you can see down here in the bottom right-hand corner, we have a cell that is just teeming with mitochondria. So all of these long sausage-shaped things are your mitochondria, and you see they're packed in there super tightly. As well as having lots and lots of mitochondria in these specific cell types that have a very high energy demand, they also have more folds in their cristae to allow more reactions to take place and therefore more ATP production. Let's talk about aerobic respiration. You should know the basic word equation that aerobic respiration requires glucose and oxygen, and it uses glucose and oxygen to produce carbon dioxide and water as our waste products, but lots and lots and lots of ATP plus some heat. So aerobic respiration requires oxygen to break down the glucose, and the process starts in the cytoplasm, but the majority of our aerobic respiration occurs in the mitochondria. So there are 3 stages that we talk about. There is glycolysis, which is the stage that happens in the cytoplasm. There is the Krebs cycle, and then there is the electron transfer chain. So in our cytoplasm, we have glucose being broken down to pyruvate. So this happens out in the cytosol, or the cytoplasm of our cell, and glycolysis produces pyruvate. So again, this is an enzyme process. So that an enzyme breaks down glucose to pyruvate. The pyruvate is then sent to the mitochondria, to the Krebs cycle, which occurs in the matrix of our mitochondria. So again, if you remember the Krebs cycle, the matrix of our mitochondria is just packed full of enzymes, and all of those enzymes are going to be involved in the Krebs cycle, breaking down pyruvate to basically produce carbon dioxide and water and lots of hydrogen ions or hydrogen atoms. So those hydrogen atoms are then used or carried to the cristae of our mitochondria, and they're sort of passed backwards and forwards. The electrons from our hydrogen atoms are passed backwards and forwards across the membrane of our cristae in what is known as the electron transfer chain. So they will be passed backwards and forwards, and in doing so, they produce lots and lots of ATP. So here we are, nice summary diagram. Glycolysis occurs in the cytoplasm. One molecule of glucose is broken down to some pyruvate, and in the process, it produces a couple of ATP. This is important because when you're talking about anaerobic respiration in an animal cell, that's what anaerobic respiration is. It's just glycolysis. So it is the first stage of respiration. Occurs in the cytoplasm, produces 2 ATP. Next, the pyruvate moves into the matrix of the mitochondria, and through a complex series of enzyme reactions that you guys don't need to know, pyruvate is then broken down to produce carbon dioxide and hydrogen atoms. And those hydrogen atoms are then moved to the cristae of the mitochondria. The cristae is where the electron transport chain happens. Those hydrogen atoms are ionized to H+, and the electrons from those hydrogen atoms are then passed backwards and forwards between the molecules on the cristae. And when they do that, the energy from those electrons is used to turn ADP back into ATP. So basically, we are refueling our ADP by using the energy from our electrons to add on an extra phosphate and turn it back into ATP. And they're able to do this very, very efficiently, producing about 36 ATP from one glucose molecule. And as a result, water is produced as a waste product. Now, you can study for years the process of aerobic respiration, and there are loads of enzymes involved in the process, but in terms of what you need to know for level 2, this summary slide will get you there. Okay, this is the level of detail that you need to know. Any quick questions about?
Just a few. What does the oxygen do in the electron chain? I know it enables electrons to be passed.
From memory, oxygen is not really involved in the electron transport chain. It's produced as a product or byproduct of the Krebs cycle. So the hydrogen atoms are the ones that get passed to the electron transport chain, and they are ionized, and the electrons from the hydrogen atoms are used to generate ATP.
Cool. And I think I might have already asked this. What are enzymes' role in photosynthesis? Did I ask that before?
Yeah, I think so, yeah. So photosynthesis, again, is a very complex process that involves lots of enzymatic reactions. So a lot of enzymes are involved in photosynthesis, but you don't need to specifically know the details.
Cool, that's it.
Okay. So anaerobic respiration, this is glycolysis. It takes place in the cytoplasm of the cell, no mitochondria are required, and it doesn't require any oxygen. Because it's basically glycolysis, it only produces 2 ATP for each molecule of glucose. It's great for producing a bit of ATP if you're in a bit of a pinch, but for most animal cells, they would much prefer to be performing aerobic respiration because they get a lot more ATP out of the process. So here we see our glucose being broken down through glycolysis to produce pyruvate. This is in the absence of oxygen, and through another process, it produces lactic acid as a waste product. And the lactic acid can be quite toxic to our cells. So in terms of the factors that affect respiration, remember, respiration is an enzyme-mediated process or an enzyme-controlled process. So all of the things that affect enzyme function are going to affect cellular respiration. If it's too cold, respiration will be slow. At the optimum temperature, you're going to have respiration happening efficiently at the ideal rate. Too hot, and you're going to start losing enzyme function because those enzymes may denature. So if it gets too hot, respiration can stop entirely. pH, again, can be involved in denaturing the enzymes. So once the enzymes denature, you're not going to have any respiration occurring. But also, we need to think about the concentration of our substrates. So yes, respiration is an enzyme-mediated process, but if we go back to think about the word equation for respiration, we know that glucose and oxygen are needed to produce carbon dioxide, water, and loads of ATP. So if the concentration of glucose is low, then respiration is going to be slow. There are going to be no reactants in order for respiration to occur. If the concentration of oxygen is low, then aerobic respiration is not going to be happening. We're going to get anaerobic instead. Okay, so shortages of glucose or oxygen will slow down respiration. Again, because it's an enzyme-mediated process, if you have an enzyme inhibitor chucked into the mix, such as cadmium or other substances that are able to prevent the enzymes from catalyzing the reaction, then you're not going to have respiration occurring. Specific to respiration, one other factor that affects respiration is demand. So in animals, their demand for energy will cause an increase in respiration up to a maximum point. So I don't think I'll have time to go through it, but I've got quite detailed notes. One of the past exam questions that we looked at was from last year's exam, which talked about a mako shark, which are capable of bursts of very, very fast swimming. So in those times where the mako shark has this burst of very, very fast swimming, suddenly their demand for energy skyrockets. Their demand for energy increases hugely, which means an increase in the rate of respiration up to a maximum point. So that is what we talk about when we talk about demand. So depending on what the animal or the organism is doing, when they have a high demand for energy, this will cause an increase in respiration. And this is the question that I was talking about there with the short-finned mako shark. It discusses where and when anaerobic respiration occur and asks you to explain the difference between them and where they occur in the shark cells, and identify where each type of respiration occurs in an animal cell. To explain why the mako shark can only carry out anaerobic respiration for short periods of time. And the reason for that is that you're going to get lots of buildup of waste products like lactic acid. And to discuss the advantages and disadvantages of the mako shark carrying out both anaerobic and aerobic respiration. So aerobic respiration produces lots and lots of ATP. All of that ATP is going to be able to feed the mako shark's muscle cells, so they're going to be able to swim really fast. However, it is a slower process than anaerobic respiration. It requires oxygen, and the oxygen needs to be transported to the cells for aerobic respiration to occur. In contrast, you've got anaerobic respiration, which will happen in the shark's muscle cells when they do have a really, really fast burst of swimming, because it's able to release energy quite quickly. Yes, only 2 ATP, but it doesn't need to worry about oxygen being able to diffuse into the cell to be used for aerobic respiration. It can happen very, very quickly, but the shark's only going to be able to use anaerobic respiration for a very short period of time before lactic acid starts to build up and become quite toxic to the cell. And I do have all of that explained on these slides here. So the slides are available. I think the link is in the webinar description, so you can have a read of that answer. As it is now 8:45, is there anything that we would like to wrap up with?
Give it 30 seconds or so just for them to type up their answers.
Yep, no worries. Been very impressed with all of the questions that people have asked. I think last year when I did a webinar, I got about 4 questions.
Just one question, or it's just photosynthesis. Well, I think that's a rather large topic.
It is, yes.
Yeah.
I do have some quite detailed slides on photosynthesis. So hopefully, if that student wants to have a look through the photosynthesis slides, hopefully that will help. If they do have specific questions, I'm happy if you, I don't know what email address they've got. If they're able to pop a question or a comment on the YouTube link, I can answer them that way, perhaps.
Is it in this PowerPoint? Or is it different?
Yes, no, the details on photosynthesis are in this PowerPoint.
Okay, this PowerPoint's the video links. I know the PowerPoint links in the description, so they can just go to the description. Someone asked surface area to volume ratio. Is that something they'll need to know?
Yes. So in terms of surface area to volume ratio, very small cells, or when cells are very small, they have a small amount of volume and a large surface area for the plasma membrane on the outside. As cells start to grow or to swell and get bigger, the volume, the amount inside the cell, increases much faster than the surface area on the outside. So what this means is you get a limit to how big a cell can grow. What limits the size of the cell is if we think about all of those diffusion processes that we talked about, facilitated diffusion as well as just passive transport and moving oxygen and other gases and things into the cell, that all needs to occur on the cell membrane. So you need to have a decent amount of surface area for all of that to occur to get all the way through the cell. So once the cell starts getting too big and has too much volume, those substances are not going to be able to diffuse effectively or efficiently into the cell to be able to feed the cell and to get all of those substances where they need to be inside the cell. So what you get is the cell will usually undergo mitosis and split into two smaller cells at that point.
Cool. I'll just quickly scroll up and check the rest. Tips and tricks on how to approach exam type questions of DNA replication and mitosis. They've asked to show a sample question from NCEA relating to mitosis and DNA replication, but don't know if we have time for that. I don't think we have time for that.
I don't. I think that is towards the end of my slideshow, so there should be an example of a sample question relating to mitosis and DNA replication. In terms of tips and tricks, you should know the processes of DNA replication, what happens and in what order. You need to be able to explain the purpose of DNA replication. It's to produce an identical copy of the cell's DNA, and that every cell needs a copy of that DNA. The purpose of mitosis is to produce two identical daughter cells that have a complete set of DNA that are able to perform the same functions as the parent cell, and that they are used for growth and for repair and replacement of damaged cells.
Cool. Hopefully that's answered that. What should you consider other than things specific in the question when answering? What are the key things to remember for excellence?
Yeah, so I guess we'll start off for achieved for most of the questions that you'll get. The first bullet point will be directing you towards what your achieved points are going to be, and they will usually be either identifying what's happening in the question, so being able to read the question and understand what's going on at a basic level, or just providing some a definition, like a definition of what is aerobic respiration, what is anaerobic respiration. For merit, you would be expected to explain those processes in detail, and then for excellence, you'd be explaining those processes in detail and linking them together and linking them to the context of the question. And that's usually somewhere down around your third bullet point. And so you'd be talking about, like as we did with the Mako shark, you'd be talking about aerobic respiration and anaerobic respiration, and what they mean for the cell in general, but also what they mean for our Mako shark in terms of how the Mako shark is able to use anaerobic and aerobic respiration effectively when it is swimming. And then you would then go on to talk about the advantages and disadvantages of aerobic and anaerobic respiration. You're comparing and contrasting those two different processes.
Cool. I think that's already been covered. Meiosis will be covered in genetic variation. I don't know what that means.
Yep.
Yes. Cool. I think someone already answered the light independent phase occurs, and someone's already answered that. So does mitosis, does the independent assortment or crossing over, or does the genetic information just move to the polar sides of the cell and that's it?
So mitosis does not involve any crossing over or independent assortment. Mitosis is just copying the cell, replicating the DNA, and dividing the cell in two. So independent assortment and crossing over are in meiosis. So they'll be talked about in genetic variation.
Cool. And just to follow up, meiosis is for the creation of the sex cells for everyone else.
That's correct. Yeah.
Including myself. I think that's all the questions. Yeah, that's all the questions for now.
Cool. Fantastic. And thank you for the lovely feedback. It's nice to hear that you guys don't think that I talk too fast and that you're finding the webinars really helpful. So thank you for that. And all the very best for your cellular processes exam. And hopefully I'll have some of you back on Sunday for, what have we got? Gene expression on Sunday, I believe.
I think so. For those that are wondering, it's in the description. So there's one of them is on Sunday night and the other one is on Monday night, I believe. And right. I'll see you all there. Right, it's 8:53 and we finish off. Yeah, it's
yep.
Cool.
Yep. Sorry, guys. Thank you.
Right.
All right.
Right. So just like again, once again, thank Amy for spending an hour and 50 minutes going over the level 2 biology cells and processes exam. I struggled to keep up at times, but hopefully I managed to answer or ask as many questions as possible. If there's any questions that you have, you can go to Study It and ask them or ask it on Study It. And if you've got any really good questions, you can save them up for the tutorial on Sunday or the tutorial on Monday. I'm sure Amy will be glad to answer them. Other than that, thank you, Amy, for spending your time, and we'll call it a day.
Fantastic. Thank you, Andrew.